Pharmaceutical drug analysis video 4 part 1.3 management. This is a page 6. Various official compendia of different countries categorically specify descriptive as well as informative details with regard to the pharmaceutical substances and formulated dosage forms produced therefore. Hence, all pharmaceutical chemicals and finished products must rigidly conform to the laid out standards in a particular country and are subjected to various checks at different levels, either by governments, state-owned drug testing laboratories, or by governments' state-approved drug testing laboratories. Official compendia for pharmaceutical substances usually include the following parameters, namely description of a drug or finished product, identification test, physical constants, assay of pharmaceutical substances, assay of principal active ingredients in formulated dosage forms, limit test, and storage condition. 131 Description of the drug or finished products. The description of a particular drug or finished products may essentially include the following details, namely brand name of a product, name of the active ingredients, strength of the active ingredient in dosage form, lot or batch number, date of the manufacturer, date of the expiry, storage condition, if any kind of the storage condition is needed, separate dosage for adults and condition. 1.32 Sample procedures and errors. To collect a representative sample form vital aspects of analytical chemistry because the samples subjected to analysis are assumed to be perfectly homogeneous and truly representative. Thus, sampling may be considered as the most critical aspects of analysis. In other words, the accuracy and the significance of measurements may be solely limited by the sampling processes, unless and until the sampling processes is performed properly. It may give rise to a possible weak link in the interpretation of the analytical results. For instance, the improper handling of a blood sample both during and after sampling from a patient prior to an operation may not only pose serious complication but also may prove fatal. A definite instruction with regard to a sampling of given materials have been duly put forward by a number of professional society, namely Association of Official Analytical Chemists, AOAC, American Society for Testing Material, ASTM, and American Public Health Association, APHA. However, a good deal of the wisdom of the analysts supported by the application of statistical results and wealth of experience may go a long way in achieving reasonably accurate and reproducible results. 1.21 Sampling procedures. Samples may be categorized broadly into 4 heads, namely gross sample, a sample that represents the whole lot and may vary from a few grams or less to several pounds based on the nature of the bulk materials. Sample a sufficiently small size of the sample exclusively for the purpose of analysis and derived from the representative gross sample, analysis sample, and aliquot or portion of the sample being subjected to actual analysis. Grab sample, a single sample usually taken at random and assumed to be representative. It is considered to be the most unreliable way to sample a material. Sampling procedure is a gross sample, analysis sample, and grab sample. Okay, the next part is for solids. Sampling of solids material are comparatively more difficult than other materials because of the following three reasons, namely variation in particle size, inhomogeneity of the material, and variation within the particle. Sampling of solids can be best accomplished by adopting the following procedures. To take 150 to 1/100th of the total bulk for gross sample, a sample that represents the whole lot and may vary from the few grams. To take larger gross sample for products having larger particle size. To sample large bodies of solid material while they are in movement to obtain aliquots representing all portion of the bulk, or to handle tissue samples. Several tiny parts of an organ may be taken and combined together. The next parts. For liquids, sampling of liquids may be carried out by following these procedures. Small heterogeneous liquid samples are first shaken thoroughly and then followed by immediate sampling. Large volume of liquids are best sampled immediately after a transfer or even pipeline after passing through a pump where it has undergone the most vigorous mixing. Large volume of stationary liquid are normally sampled with a excuse me سگوریا اینجا ببینم بدو اینجا ببینم بدو اینجا بدو دم درس بخون ساکت excuse me دم درس میخونم. Oh my god, sorry that they are disturb you. They are very good pets. They are really good pets. They are very good pets and they allow me to make a video. They are good pets. This is Juji, good girls. You are good girls. I know you are good girls. Okay. Large volume of stationary liquid are normally sampled with a TIFF sampler, that means device for collecting a liquid at different levels. Samples are best drawn with a TIFF sampler at various depths diagonally instead of vertically down, so as to have a better cross section of the bulk liquid. And my pet also wants to learn. Okay, either separate liquids of liquid may be analyzed individually and results combined duly, or the various liquid may be first combined into one gross sample and the replicate analysis carried out. However, the later method is preferred for obvious reason, since the analysis shall have a better hold on the accuracy and precision of the analysis. For sampling of biological fluid, the time factor is of is of utmost importance and hence should be performed by qualified pathologists attached to clinical laboratories under adequate supervision. A few specific examples are stated below. A 24-hour urine sample collection are usually more reliable than single specimen. A sample for blood sugar analysis is more reliable in a fasting patient. A sample of cerebrospinal fluid CSF from the vertebral column by lumbar puncture in patients having suspected pyogenic meningitis. The next part for gases, a grab type gas sample is usually satisfactory in certain cases. For example, a breath sample may be collected by allowing the subject to blow into an evacuated bag. Persons driving automobile under the influence of alcohol on highways during festive seasons. Auto exhaust, auto exhaust may also be collected in large evacuated plastic bags to monitor the pollution by vehicles run by gasoline, diesel, CNG in cities and metropolis. The next parts errors. The famous edge. Okay, I take another videos for the errors.